Okay, good morning students. Please wait for a minute. Okay, till the speed stabilizes. Okay, let's start our lecture. Okay, speed is stabilized now. Okay, as we discussed yesterday about the fermentative patterns using various fermentation pathways. Okay, today I am going to discuss about the Archaebacteria, in which your course includes two of them. One of them is a methanogen, and the another one is an halobacterium. Uh, the energy metabolism, basically how they produce energy and how carbon assimilation occurs in methogens, also and also how they produce methane. Okay, there are three different types of methanogenic pathways, and how photophosphorylation occurs in the halobacterium. Okay, methanogens, as you all know, we have studied the methanogens in your semester 3 under archaea. Methanogens are simply those organisms, those bacteria or archaea which produce methane gas, and they are commonly found in swamps, lakes, sediments, intestinal tracts of animals, or even anaerobic sewage digesters. Okay, and also one more important point, that most of them are anaerobic ones, and it results from the activity of highly specialized group of bacteria that convert fermentation products formed by another anaerobes, okay, products such as carbon dioxide, hydrogen, formate, acetate to methane, or methane and carbon dioxide. And since methane gas is sparingly soluble in water, it escapes from the anaerobic environment, hence may be aerobically oxidized by the members of other bacterial group, okay, the methophiles, and usually interferes between the anaerobic and aerobic conditions also. And methanogens are consequently terminal members of the anaerobic food chain. Okay, in some anaerobic environments, example sewage digesters and some of the sanitary landfills, and methanogenesis occurs at a rate that makes collection and compression of this principal component of natural gas economically feasible. I don't know if there is any landfill in India which produces methane gas, okay, but there are landfills in US where with the help of landfill they have they are using such a mechanism that they can collect methane gas and use it for the purpose of the power generation. Okay, I don't know if there is any landfill which have such a facility to collect methane. Okay, so now moving forward with the methanogens. Okay, these are the two examples. There are also various examples, okay, photographs of two examples, and one is a SEM image, that is scanning electron microscope image, and another one is TEM image, which is transmission electron microscope. And Methanobrevibacterus smithi given here, and the Methanococcus burtoni, okay, given here. Okay, so how methanogens and even some of them other than methanogens can also produce methane, okay, but they are not archaea, okay, they are not archaea. And there are various other places where you can get an idea of methanogens, but the problem with the methanogen study lies with the isolation part, okay, lies with the isolation part. Why isolation part is a problematic? The reason is very simple, okay, because not even a single methanogen has been isolated till now, okay, not even a single methanogen is been isolated till now, okay, due to which very less amount of the study has been done on the methanogens. Whatever the study has been done is because of the metagenomics, okay, is because of the metagenomics. And some of the methanogenic bacteria, okay, are basically classified into Methanobacterium, Methanococcus, and Methanosarcina, and one of them is spiral also, Methanospirillum, okay, and they are a special group of bacteria which are under Archaebacteria, okay, but as compared to bacteria, they lack a typical peptidoglycan structure in their cell, okay, they have a pseudomurine type structure, okay, they have a pseudomurine in their structure, okay, and the peptide sheet is found in Methanospirillum, okay, and the cell wall of Methanosarcina barkeri consists of a polysaccharide which is basically composed of uronic acids, neutral sugars, and even amino acids, okay. As I told you, uh, we are still not able to isolate a colony of a methanogen. Now the photograph given here, okay, methane is collected in a funnel from a swamp sediments where it was produced by methanogens and then ignited in a demonstration experiment at Woods Hole, Massachusetts, USA. Okay, swamps, do you know what are swamps? Swamps are basically waterlogged areas, okay. Swamps are basically waterlogged areas, okay. Now moving forward with the basic mechanism, okay, with the basic mechanism, okay. Now nutrients may be in the form of biomass, polysaccharide, proteins, fats, etc., okay, and this biomass basically this biomass will be converted into hydrogen gas, carbon dioxide or acetate, or propionate, butyrate, succinate, alcohols. This is done by the fermentative bacteria, okay. There are various kinds, okay, there are various kinds of fermentative bacteria, okay, it may be any of the fermentative bacteria, okay. Now the conversion, okay, one are purely methanogenic bacteria, okay, which produce methane directly from the hydrogen and carbon dioxide, okay, and one goes through from propionate, butyrate or succinate alcohols which are known as basically acetogenic bacteria, okay, which are known as acetogenic bacteria, okay, which produce hydrogen, carbon dioxide, acetate, okay. This is done by acetogenic bacteria, this process, and finally this process is carried out by methanogens, okay, this process is carried out by methanogens. And as we have studied in our semester 6, the methanogens are basically strict anaerobes and exposure to air is very little to them, okay, is very little to them. They neither contain catalase nor superoxide dismutase, and their extreme sensitivity to oxygen is a cause of scarcity of the existing knowledge, okay. We do not know the exact how and why they are sensitive to oxygen, because the problem lies in the isolation, okay. And most of the methanogens are isolated and kept in pure culture up now can use hydrogen as hydrogen donor. But how are they isolated? Okay, they are isolated from the remains of various animals, okay, and they are always set up in a mixture of cultures. They are always set up in a mixture of cultures. And methogenic bacteria are the last link in the anaerobic chain, okay. The process is very simple. I am repeating the process again. Bacteria that ferment various polysaccharides such as cellulose, okay, will be converted into succinate, propionate, butyrate, lactate, acetate, alcohols, carbon dioxide, hydrogen gas, etc. Okay, and the second one, which is a acetogenic bacteria here, okay, which is an acetogenic bacteria, ferment these products to acetate, formate carbon dioxide and hydrogen, and these products are the substrate for the methanogenic bacteria. And methanogens occur basically in close association with the hydrogen producing bacteria. Whenever there has been a trial done to isolate a methanogen, okay, there are always a combination of cultures have been reported, okay, and one of which is basically hydrogen bacteria, okay, that is hydrogen gas producing bacteria. In the microhabitats, hydrogen is rarely liberated in the gaseous form. Rather, the excreted hydrogen dissolved in the medium is directly assimilated by the methanogenic bacteria, okay, and methane bacteria are able to activate hydrogen and couple its oxidation to reduction of the carbon dioxide. Since they can synthesize all their cell substances using carbon dioxide as their carbon source, their mode of life is evidently regarded as chemoautotrophs. Okay, so methanogens are basically chemoautotrophs, okay, using carbon dioxide as a carbon source, okay, and hydrogen is used as an acceptor for the production of methane, okay, and the mode of respiration is also known as carbonate respiration. Now the factors that determine which of the two groups basically will predominate are not well understood, okay, and the uptake systems for acetate and hydrogen, and that is higher in the sulfur reducing bacteria than in the methogen, is thought to be contributed. It's just an idea, okay. There are basically different representative genera, okay, as I told. Now moving forward with the unique factors which have been identified till now in the case of methanogens. And methanogens contain several cofactors not found in the other bacteria, okay, of which three of them are methanoptene, methanofuran, next one is coenzyme M, are the carriers of the carbon unit during the reduction of carbon dioxide to the methane, and probably factor F420 functions as an hydrogen carrier. In these reductions, and the factor F430 is an unusual nickel tetrapyrrole and is the prosthetic group of methyl coem reductase, and which is basically the last enzyme of in the reduction pathway. And some methanogens also require coenzyme M as their growth factor, okay. Now the novel coenzymes which are basically identified in the case of methanogens, are first one methanofuran, it acts as a primary carbon dioxide acceptor, contains a furan ring and an amino acid nitrogen atom that binds to the carbon dioxide, okay. And the next one, okay, I will show you the structure in the next slide, Next one is tetrahydromethanoptene, also known as H4-MPT in the short. It resembles somewhat to a folic acid. Next one is a coenzyme M, known as COM is in a short. Okay, it is a simple chemical compound. Okay, that is basically a 2 mercaptoethane sulfonic acid. Okay, I am repeating the word again, 2 mercaptoethane sulfonic acid, and it has a reactive group which is mercapto group. Next one is coenzyme F420. It carries electrons and hydrogens. Next one is a coenzyme F430. It is a nickel containing tetrapyrrole, and this coenzyme acts as a cofactor for the enzyme methyl-CoM methyl reductase. Okay, so it basically acts as a coenzyme. Okay, acts as a basically cofactor for the enzyme methyl-CoM methyl reductase. Okay, I am giving you all half a minute to write down the names of the coenzyme. Please write down these names, because it will be easy for you to understand the pathways. Okay, because it will be easy for you to understand the pathways. Giving you all a half minute. Just write down the names of the coenzymes. Repeating the name of the coenzymes again: methanophuran, MFR in a short, tetrahydromethanoptene, H4-MPT in a short, coenzyme M, COM for the short, coenzyme F420. Next one is coenzyme F430. Moving forward with the next slide with the structures of the coenzymes. Okay, the first structure is of a methanophuran. Okay, you need not to draw these structures right now. Okay, I will share you the link of the PowerPoint presentation from which you can draw the structure. The first structure is basically methanophuran. Here is the place where transfer occurs in the case of methanophuran, and this one is a methanoptene. Okay, this one is a coenzyme M and coenzyme F430. Okay, and these are the coenzyme which function as a carbon carriers, which function as a carbon carriers. Okay, repeating the name again: methanophuran, methanoptene, coenzyme M, and coenzyme F430. Next one is basically coenzymes that can function as electron donors. Okay, of which one of them is coenzyme F420, and another coenzyme is coenzyme B. Okay, another one is a coenzyme B. Okay, now this one are the photograph. Okay, under fluorescence microscopy, where A is basically an autofluorescence in the cells of methanogen Methanosarcina barkeri due to the presence of a unique electron carrier F420. A single cell is about 1.7 micrometer in diameter, and the organisms were made visible by the excitation with the blue light in the fluorescence microscope. And another photograph to the right, okay, which is a B1, where F420 fluorescence in the cells of the methanogen Methanobacterium formicum, a single cell is about 0.6 micrometer in the diameter. Now moving forward with the process, okay, till now about 5 different phylogenetic orders of the methanogenic groups have been identified, okay, and of which I am going to discuss is basically according to the electron donors they use and compromise. Okay, they are divided into 3 groups based on their electron donors they use and they comprise of. Okay, so first one is an hydrogenotropic methanogen. First one is a hydrogenotropic methanogen. Second one is a methylotrophic methanogens. Third one is an acetic-lactic methanogens. And starting with the first one, hydrogenotropic methanogens. Okay, as the name itself says, they are going to use hydrogen. Okay, they are going to use hydrogen. Okay, for you can clearly see in this reaction where the majority of the methanogens grow on the free energy available from the reduction of carbon dioxide to the methane using hydrogen as an electron donor. They are also referred to as chemolithotrophic methanogens. And they are also able to use formate and carbon monoxide in addition to the carbon dioxide and hydrogen. They are oxidized to carbon dioxide before being reduced to the methane. And some of the chemolithotrophic methanogens oxidize ethanol, propanol to butanol completely or partially using carbon dioxide as the electron acceptor. And some of the examples of the hydrogenotropic methanogens include Methanobacterium, Methanoculeus, Methanogeum, Methanolecinia, Methanospirillum, Methanocorpusculum. Okay, this one are the examples of the reactions where first one which consists of a formate, okay, another which consists of a carbon monoxide. Okay, Okay, now moving forward with the mechanism of the hydrogenotropic methanogens. Okay, where methanogenesis from carbon dioxide and hydrogen, the carbon forming methylene, and from methylene methyl will be formed, which is basically again attached to the methanoptrene. Okay, with the help of the methyl transferase. Okay, it now gets attached to the coenzyme M.S complex. Okay, and this is how it works. COMSH complex gets involved in the reaction with this methyl one. Okay, this methyl which is in combination with the methanoptrene, where methanoptrene is released, forming a methyl complex with the coenzyme M.S complex, and this methyl group will be reduced to the methane. At the same time, a sodium motive force will be generated. For the production of the ATP. Okay, sodium motive force will be generated for the generation of the ATP. At the same time, coenzyme B, okay, complex with the HS will be converted into coenzyme, will be joining the coenzyme M for the complex. Okay, this is where a ferredoxin cycling occurs. Okay, now where we clearly see where ferredoxin, which is in reduced form, in the first reaction gets oxidized, and this oxidized will be again will again combine with the hydrogen. This is the point where hydrogen will be added. This is the point where hydrogen will be added and ferredoxin will be reduced. Okay, and at this point again, the hydrogen is being also added. Okay, hydrogen is also being added again. Okay, now I am moving forward with the next group, which are methylotrophic methanogenesis, in which methanol compounds such as methanol, methylamines are basically used as a substrate by methanogens, and some of the examples include order methanosarcinales, except those of genus Methanocita, and the strains of methanolobus can also use methyl sulfide. And one such reaction I have given here. Okay, now this methyl compound methanol will be converted into methane, carbon dioxide and water, and the methanotrophic methogens of the genus Methanosphaera use methanol only in the presence of the carbon dioxide as a electron acceptor. Now moving forward with the mechanism of methanogenic methanogenesis. For the growth on methanol, most of the carbon in the methanol is converted into methane, and the smaller amount will be converted either to carbon dioxide or via formation of acetyl CoA, and which is assimilated into cell material, Now here is the case where methanol will be transferred to coenzyme M. Okay, and from this coenzyme M, methane will be formed, and this process is of a methanogenesis, and the reducing power, okay, which is being utilized to produce methanol to the methane. Now again moving towards this side, with methanoptene will join the methanol, okay, after the release of OH, okay, and with the help of the methanopurine, reducing power is generated by oxidation of methanol to the carbon dioxide. Okay, methanol to the carbon dioxide, you can see clearly in this reaction. Okay, you will clearly see in this reaction, okay, where methanopurine basically in with the combination of this carbon group gets formed into carbon dioxide, okay, where two hydrogens will be released, and then the next reaction, okay, where two hydrogens will be released, and then dehydrogenase enzyme CO dehydrogenase will be added, okay, and this will be converted directly into the formation of the acetyl CoA for the biosynthesis. Okay, this will be converted into acetyl CoA for the biosynthesis. Okay, now moving towards the third group, acetylclastic methanogens. Where they are use acetic acid, for their production of the methanes. This includes strains Methanosarcina and Methanosarcina, and the former you only use acetate and grow in chains. And the strains of Methanosarcina are most versatile in terms of their substrate, and they can use all known methanogenic substrates except secondary alcohols. Okay, this one is a representative reaction where an acetic acid will be converted into carbon dioxide and methane. Now moving forward with the mechanism of the acetylclastic ones. Okay, acetate is basically split into methane and carbon dioxide, and also in this case also CO carbon monoxide dehydrogenase enzyme is also involved. Okay, now you can clearly see here ATP will be used, coenzyme will be added, forming a acetyl-CoA, which may either undergo biosynthesis, or may enter for the production of the methane with the help of carbon monoxide dehydrogenase complex, with the help of the enzyme carbon monoxide dehydrogenase complex, where it will be split into two ones, okay, splitting of acetate, okay, in which one methyl group will be formed, another case a CODH complex will be formed, from which directly carbon dioxide will be generated, and the water molecule basically, okay, which is basically added here, okay, and the two hydrogens will be transferred here, okay, for the generation of the proton motive force. At the same time, this methyl group gets attached to the coenzyme M, and finally from this methane will be produced, okay, methane will be produced, okay. Now moving forward with the proton motive force generating, okay, how proton motive force is generated. I have just considered a local mechanism with the help of enzyme hydrogenase, one of the common mechanism, okay. Now you can clearly see F420, which is basically reduced form, okay, where F420 will be converted into F420 reduced, and two hydrogens, where two hydrogens pass through the cell membrane and forming a hydrogen gradient, okay, and this is how proton motive force is generated. Using ferredoxin carriers, F420, okay, or F420 coenzymes, okay. So now the carbon metabolism, okay, how metabolism of the methanogens work is almost similar to that of the other autotrophic bacteria, where most of the methanogens grow well with the carbon dioxide as sole carbon source. Thus they are basically autotrophs. The pathway by which carbon dioxide is incorporated into the cell material is uncertain. It is probably similar to the carbon monoxide pathway found in some of the sulfur reducing bacteria and some of the Clostridia also. And the enzyme assay and the patterns of the labeling of the cell constituents of the radioactive carbon dioxide rule out their other known pathways of the autotrophic carbon dioxide fixation also. The ribulose diphosphate pathway and the reductive tricarboxylic acid cycle are basically also used. However, one of the two distinctive reactions of the reductive TCA cycle, namely the reductive carboxylation of acetyl-CoA, is an important step in the fixation of the carbon dioxide. And the same data rule out the two pathways by which methyllophiles use C1 units are reduced to the carbon dioxide. And acetate is an important precursor of the cell carbon in all the methanogens. However, the distribution of its level differs among the species, and suggesting that the various group of methanogens may have quite different patterns of the intermediary metabolisms. They have a variety of metabolism patterns, okay. This one is just an assumption. This pathway given is just an assumption how carbon metabolism occurs in methanogens, where you can clearly see group 1, okay, which is basically a bold line, okay, bold line given here, and group 2 simply black thin lines here, okay. There are chances, okay, how biosynthesis or carbon metabolism is basically present in these scenarios, okay, where acetyl-CoA, for example, as you all know, okay, will be converted into pyruvate, oxaloacetate, malic, malate, fumarate, succinate, alpha ketoglutarate, etc., and finally forming a glutamate, okay. Another one is acetate, citrate, isocitrate, and glutarate, and finally glutamate. Okay, this one process is basically biosynthetic pathway of the methanogens. Okay, for production of the various amino acids. Okay, now moving forward with the halophiles. Now moving forward with the halophiles. Okay, I think I will continue halophiles tomorrow. Okay, halophiles I will continue tomorrow. Okay, everyone please mark your attendance.